Hello, and welcome to this special sleep learning study guide on my channel. I know when I'm worried about studying, it's more difficult to sleep. That's why I'm creating this relaxing video to help you learn and ease your mind. Today we're diving into the basics of biology to help you prepare for that upcoming test. It's important that you study while you're awake too, but this session is designed to assist you in absorbing essential concepts while you sleep and ease your mind. You're gonna do great. You're gonna remember all the information you studied, and maybe you'll have cool science dreams with this meditation. So find a quiet space, get comfortable, and let's enhance your study experience through a sleep meditation. Before we begin, start by taking a few deep breaths. Inhale positivity and exhale any stress or tension. Feel your body sinking into a comfortable position. Clear your mind and relax. Now, let's prepare your mind for effective learning as you drift off into a peaceful sleep. Imagine a serene place, maybe a peaceful garden or a quiet forest. Picture yourself there, surrounded by a calming atmosphere. Feel the tranquility surrounding you, making it the perfect setting for focused learning. As you continue to breathe deeply, visualize a warm golden light surrounding you. With each breath, this light becomes more vibrant, relaxing your mind and preparing it for the absorption of knowledge. Now that you're in a state of deep relaxation, let's delve into the fundamentals of biology. We're going to talk about cells today. I'll provide you with key concepts and definitions that will support your understanding of cell biology. Biology is the study of living organisms and their interactions with the environment. It encompasses various subfields like genetics, ecology, and cell biology, which is what we're going to talk about today. Cells are the building blocks of life. Imagine them as tiny, intricate factories working together to sustain life. You might remember a little diagram of a cell. I want you to imagine that in your head as we identify each part of the cell and how it works. Imagine the cell surrounded by a protective barrier called the cell membrane. Imagine the little ball with the cell's outer skin, the cell membrane, controlling what goes in and out of the cell. The cell membrane is made up of a phospholipid bilayer, which you can envision it as a flexible double-layered sheet made up of phospholipid molecules. Each one of these molecules has a hydrophilic, which is water attracting, head, and hydrophobic, water repelling tails. You might remember that these phospholipids have a circle with two little squiggly lines coming off of it. The squiggly lines are the hydrophobic tails because they don't like touching water. They naturally are inside the stacked layer of phospholipids because they don't want to touch the outside on either side of the cell, which has water. So the hydrophilic ball is touching the water outside and inside the cell. So we have two sheets of these, which is the cell membrane. If anything wants to get in or out of the cell, it has to get through the cell membrane or the phospholipid bilayer. It's going to need a little tunnel to travel through the cell membrane. That's why stuck through the cell membrane or stuck through the phospholipid bilayer, there's little tunnels, and we call these tunnels little membrane proteins. They decide what gets to enter or exit the cell. They stick out through the membrane like a little tube. These are all along the phospholipid bilayer. Imagine two layers of phospholipids, then a little tube along it, allowing different molecules in or out. These are the membrane proteins. Now, let's go through the membrane protein and pass the phospholipid bilayer into the cell. If we were bacteria or a virus trying to force our way in, we might get eaten up by the lysosome in the cell. The lysosome does this, but its main duty is to help clean up the cell. It has digestive enzymes, so it can eat all the old, worn-out parts of the cell that need to be replaced. The lysosome is kind of like the cleanup crew. Just inside the cell membrane are these little noodly things. You might remember from a diagram you've seen before. These little noodly things are called the cytoskeleton. The cytoskeleton is made of microtubules and microfilaments. I remember the cytoskeleton is made of microtubules and microfilaments because those words kind of sound like noodles to me, like different kinds of noodles. The cytoskeleton helps keep the shape of the cell, and it's extra support for the membrane. So now that we're inside of the cell, we're going to swim across in the cytoplasm. The cytoplasm is all over the cell's inside. It looks like a mix between liquid and jelly. Inside of the cytoplasm are all the organelles in the cell's nucleus. The cytoplasm is made of water, protein, and different salts. So we're going to go through the cytoplasm, and the big ball in the middle is called the nucleus. The nucleus is surrounded by a double membrane nuclear envelope. You can think of this as kind of similar to the cell membrane. Picture this envelope on the nucleus as a protective barrier with nuclear pores. Nuclear pores kind of look like little holes all over the nucleus. So imagine a ball, little holes all over it. The little holes, called the nuclear pores, decide what molecules can enter or exit the nucleus. Now, the nucleus has DNA in it. And you might know what DNA looks like because it has a double helix structure. Now, the inside of the nucleus has these threads wrapped around beads. So imagine a thread wrapped around a bead, and then another bead, and another bead. Now, imagine that the thread is DNA, and the bead that the DNA is wrapped around are histone proteins. Now, the reason the DNA is wrapped around the histone protein is because there's a bunch of DNA and we need to condense it so it fits inside of the nucleus. So the histone proteins condense the long strands of DNA to fit inside the cell. Now, when the DNA is wrapped up like this with the beads, we call the whole thing chromatin. So, you can imagine that the bead is the histone protein, the thread is DNA, and the whole nucleus is chromatin. When there's 8 beads of histone proteins, they kind of clump together, and the thread of DNA gets jumbled up even more. So when 8 beads are together, the mass of those 8 beads is called a nucleosome. So imagine a thread with a clump of nucleosome. Now, deeper into the nucleus is another ball, and that ball is the Nucleolus, which is just a smaller sphere, and instead of DNA, the Nucleolus has mostly RNA. It can help make ribosomal RNA. Imagine tiny workers inside the nucleolus, which again is inside the nucleus. These workers are like gene readers, and we call them RNA polymerase. RNA polymerase read special instructions called rRNA genes to create rRNA molecules, kind of like assembling furniture. Now, picture that these rRNA molecules are joining forces with some helper friends, which are the proteins. So, the rRNA molecules work together with proteins to create the pre-ribosomal particles. These particles are like little structures that eventually are going to build a fully functional ribosome. And ribosomes help build more things in the cell. So, in simpler terms, inside the nucleolus, workers read instructions to make special parts. And these parts team up with proteins to form building blocks that will eventually become ribosomes in the cell. The ribosomes are kind of like little construction workers, but they have to be built first. So all the stuff that happens in the nucleolus and all of the DNA that's in the nucleus, all of that's in the nucleus. Now, let's go to the outer membrane of the nucleus, which is connected to the endoplasmic reticulum. The endoplasmic reticulum is sometimes shortened to just be ER. There's the smooth ER and the rough ER. The endoplasmic reticulum connected to the nucleus is the bendy looking thing you see in diagrams. It's just outside the nucleus and it helps with making proteins. The process of making proteins takes place on the rough endoplasmic reticulum, or the rough ER. It's called the rough ER because it's studded with ribosomes. So it's the bendy looking thing with little dots all over it. So, like we said before, ribosomes are like little construction workers. They facilitate protein production. So you can imagine the rough endoplasmic reticulum as a busy workshop inside the cell where workers, who are the ribosomes, are building important tools, the proteins. And they're building these proteins based on the instructions they get from mRNA. So imagine this process inside the nucleus, instructions are created, the mRNA, that contain the information needed to build the proteins. The instructions, the mRNA, are carried to the rough ER, which is called rough because it has ribosomes on its surface. So inside the rough ER, the ribosomes read the mRNA and start building proteins piece by piece. As the proteins are being built, they go through some final touches and adjustments inside the rough ER. Now, once the proteins are ready, they're packed into transport vesicles. And the transport vesicles deliver the proteins to another part of the cell. So the transport vesicles move from the rough ER to another place called the Golgi apparatus, where the proteins receive some final packaging and are sent to their specific destinations. The Golgi apparatus is sort of like a quality control factory. It's a stop along the way in the protein's final destination. So, inside the Golgi apparatus, workers carefully inspect and make final adjustments to the protein to make sure everything is in order. And then they add little labels, which we call molecular tags. So that way the proteins know where to go in the body and in the cell. So then after all that's done, the proteins are repackaged into new boxes, which are new transport vesicles. And they're delivered to their designated locations in the cell. Finally, these new transport vesicles move towards the cell's membrane, where they either release their contents outside of the cell or deliver the proteins to specific parts in the cell that need them. So, the rough ER and the Golgi apparatus are always talking to each other. The Golgi apparatus is just outside the rough ER, and it also kind of looks like a bendy structure. Sometimes in drawings or diagrams we see a mitochondria in the cell. They look like little 3D ovals in diagrams usually, but they can have different shapes. We'll think of them as ovals for now to keep things simple. Now, in diagrams, there's usually only one mitochondria, but really, there's hundreds or thousands of mitochondria inside of a cell. The mitochondria makes ATP, which is the energy source of our cells. ATP is a nucleotide, and it stands for adenosine triphosphate. It provides all the energy our cells need and supports different processes in our cells. A lot of people talk about ATP like it's this currency of the cell because it gets traded around a lot. So that's why people say the mitochondria is the powerhouse of the cell, because it's making all the ATP, which is super important to all the processes that keep us alive and keep us moving around. To understand how ATP is made in the mitochondria, you have to understand respiration. So, the mitochondria is more complex to explain, in my opinion, because it's almost like its own cell. There's a lot going on in the mitochondria, but to simplify it, think of the mitochondria as a little power plant inside of the cell. Imagine the mitochondria as small, elongated shapes with 2 layers of membranes. The inner membrane has folds, making it look like the surface of a crumpled paper. And these folds increase the space for important reactions. Now, the mitochondria's main job, of course, is making energy, the ATP. Picture mitochondria as factories where chemical reactions happen. Now, there's different pathways inside this power plant. Some of the pathways are named the Krebs cycle, and some of the pathways are the electron transport chain. So, we're going to talk about the Krebs cycle. Imagine a busy hub inside the power plant, the mitochondria, where special molecules go through a series of reactions, creating energy carriers called NADH and FADH2. The Krebs cycle is also known as the citric acid cycle. The Krebs cycle is a series of chemical reactions that occur in the mitochondria of cells. Its main purpose is to generate energy in the form of ATP and produce electron carriers for the electron transport chain. So the Krebs cycle begins when a molecule called acetyl coenzyme A, which we can shorten to acetyl-CoA, combines with a 4-carbon compound called oxaloacetate. So when acetyl-CoA combines with the 4-carbon compound oxaloacetate, forming a 6-carbon compound known as citrate, citrate undergoes a series of reactions where carbon dioxide is released. And when carbon dioxide is released, the molecule is gradually transformed. These reactions release energy that is captured in the form of electron carriers like NADH and FADH2. The energy release in the form of electron carriers is used to make ATP. GTP is also produced, and it's similar to ATP. At the end of the cycle, the original 4-carbon compound, oxaloacetate, is regenerated. This compound can then combine with another acetyl-CoA to start the cycle again. So, in simpler terms, the Krebs cycle takes the 2-carbon acetyl-CoA molecule processes it through a series of reactions, and then in the process releases energy stored in the form of ATP and electron carriers. The cycle not only produces energy, but also provides electron carriers that then feed into the electron transport chain, which is a subsequent step in cellular respiration that further contributes to ATP production. The electron transport chain, which is also in the mitochondria, is a chain of proteins in the inner membrane of the mitochondria. As electrons move through these proteins, it's creating a power gradient, similar to water flowing through a dam. Now picture a tiny turbine in the inner membrane. The turbine is ATP synthase. It uses the energy from the electron flow to make ATP, the cell's energy currency. So we can think of the mitochondria as a power station. They turn the chemical energy and nutrients like glucose into usable energy stored in ATP. It's like converting raw materials into electricity that powers different parts of the cell. Now that we've gone over some of the more challenging parts to explain in the cell, let's zoom out again and look at the whole cell and just give a little overview of all of the parts. So, like we said, we have the cell membrane, the outer boundary of the cell, a flexible layer of lipids and proteins. The cell membrane regulates what enters and exits the cell and provides protection and support. Then we have the cytoskeleton, which helps maintain the structure of the cell and is made of microtubules and microfilaments. Then we have the cytoplasm, which is a gel-like substance filling the cell where organelles are inside of it. The cytoplasm helps support and protect the organelles and facilitates cellular processes. Then we have the nucleus, which is the control center of the cell. And most of the time, it's right in the middle of the cell. The nucleus has all the genetic information and directs cell activities. The genetic information is called DNA. On the outside of the nucleus is the nuclear envelope, which is the double membrane surrounding the nucleus. It controls the passage of materials in and out of this nucleus. Inside of the nucleus is the Nucleolus, which is involved in ribosomal RNA synthesis. Ribosomes are small particles made of RNA and protein, and they help with protein synthesis. The endoplasmic reticulum is an extensive network of membranes which can be rough or smooth. The rough ER has ribosomes. The rough ER synthesizes proteins, and the smooth ER is involved in lipid metabolism and detoxification. The Golgi apparatus are kind of like stacks of flattened sacks, like pancakes almost. The Golgi apparatus modifies, sorts, and packages proteins for distribution. Both the rough ER and the Golgi apparatus use transport vesicles to send proteins to different places. The mitochondria is a double-membraned organelle with inner folds called cristae. It produces ATP through cellular respiration involved in various cellular processes. ATP stands for adenosine triphosphate, and it's like the currency for the cell. Lysosomes are membrane-bound vesicles containing digestive enzymes. They break down waste materials, cellular debris, and foreign substances. We haven't talked about centrosome or centrioles yet, but they kind of look like little pasta, little tubes in the cell. And centrosome and centrioles are microtubule organizing centers with paired cylindrical structures. They're involved in organizing microtubules during cell division. The flagella and cilia are kind of like tails or whip-like extensions on the outside of a cell. They have little tails. Flagella are longer and cilia are shorter. They propel the cell or moving substances along the cell's surface. Now let's talk about cell division. Cell division is the process in which a parent cell divides into two or more daughter cells. Each of these cells can then further divide, and then those cells can divide, and so on. This ensures the growth, development, and repair of multicellular organisms. There are two main types of cell division: mitosis and meiosis. So, mitosis is a form of cell division that occurs in somatic cells. Somatic cells are non-reproductive cells. So, mitosis results in the production of two identical daughter cells, each with the same number of chromosomes as the parent cell. The stages of meiosis are interphase, prophase, metaphase, anaphase, and telophase. So the first stage of mitosis is interphase. Before mitosis begins, the cell undergoes a period of growth and preparation called interphase. During interphase, the cell duplicates its DNA through a process called DNA replication. So it's getting ready to have its DNA in two different cells. Then after interphase is prophase. In prophase, the chromatin condenses into visible chromosomes, which are the X-like structures you see. So usually they're not in that X structure. They're just kind of bundled up chromatin. So in prophase, chromatin condenses into visible chromosomes. The nuclear envelope breaks down and spindle fibers made of microtubules begin to form and extend between opposite poles of the cell. So the cell is getting ready to pull apart, and the DNA is getting ready to go into each one of the new daughter cells. After interphase and after prophase is metaphase. In metaphase, chromosomes align at the cell's equator, right in the middle, forming the metaphase plate. So imagine two cells that are kind of squished together, but like in the midst of breaking apart. The chromosomes are right in the middle, and spindle fibers attach to the centromeres of each chromosome. After metaphase is anaphase. In anaphase, centromeres split, separating sister chromatids. Spindle fibers contract, pulling chromatids to opposite poles of the cell. After anaphase is telophase. In telophase, chromatids reach opposite poles and decondense back into chromatin. Nuclear envelopes reform around each set of chromosomes. The cell undergoes cytokinesis, the division of cytoplasm, resulting in two distinct daughter cells. These are all the stages of mitosis. We have interphase, prophase, metaphase, anaphase, and telophase. The other kind of cell division is called meiosis. Meiosis is a type of cell division that occurs in reproductive cells, leading to the formation of gametes, which are sperm and egg cells. It involves two successive divisions, resulting in 4 non-identical daughter cells, each with half of the number of chromosomes as the parent cell. There are 2 stages of meiosis: meiosis 1 and meiosis 2. Meiosis 1 has prophase 1, metaphase 1, anaphase 1, and telophase 1. And meiosis 2 has prophase 2, metaphase 2, anaphase 2, and telophase 2. So, in meiosis 1, in prophase 1, chromosomes condense and the homologous chromosomes exchange genetic material in a process called crossing over. In metaphase 1, homologous chromosomes align at the metaphase plate. After this, in anaphase 1, homologous chromosomes separate and move to opposite poles. In telophase 1, chromosomes arrive at the poles and the cell undergoes cytokinesis, forming two haploid daughter cells. After this is meiosis 2, which starts with prophase 2. In prophase 2, chromosomes condense again into two haploid cells. Then, metaphase 2 begins, where chromosomes align at the metaphase plate in both cells. After this is anaphase 2, where sister chromatids separate and move to opposite poles. Then, telophase 2 is where chromatids reach the poles, the nuclear envelope reforms, and cytokinesis occurs, resulting in a total of 4 haploid daughter cells. So, in summary, mitosis leads to the production of genetically identical daughter cells, while meiosis produces non-identical haploid cells with half the chromosome number for sexual reproduction. Both processes are crucial for the maintenance of an organism's structure and genetic diversity. Now that we've gone over some of the basics of biology and cells, let these concepts gently embed themselves into your memory. Your subconscious mind is an incredible learner, and as you sleep, it will absorb and retain this valuable information. Thank you for joining us on this sleep learning journey. If you need to rewatch this video, you may do so. If you found this session helpful, be sure to like, share, and subscribe for more study guides and sleep learning content. Wishing you the best of luck on your upcoming test or exam or whatever you need this information for. I hope that you wake up feeling confident in your newfound biology knowledge. Now go to sleep.